Let's study about immunotherapy for cancer. When tumor is formed, there are special environment so-called tumor microenvironment is formed. In this unique structure, there are multiple states of tumor cells and also various immune cells are involved. When the cells goes wrong and the cells becomes cancerous, our immune cells can attack them and then remove them from our body. That's why the chance of getting tumor is relatively low. But sometimes the defense line is broken and then cancer cells start to grow and then grow into big chunk, so-called tumor is formed. In this case, the initiating cell, we believe there are tumor initiating cell or so-called cancer stem cells. And then differentiated tumors, they fill up to the big chunk of the structure of tumors. Once the tumor size becomes quite big, then the problem is the excess of a nutrient or oxygen inside the tumor is becoming very difficult. In this case, the tumor attracts those new growths of the blood vessel cells, and then this process is called new blood vessel formation or angiogenesis. Through this process, immune cells, even though they failed to defend the first line of the cancer growth, still they are trying to regulate those tumors. And those are macrophages and also precursors of macrophages, so-called myeloid the derived suppressor cells, and also the supporting cells, dendritic cell, and natural killers, and B cells and T cells. Not only those immune cells, in this big chunk of a tumor, there are the changed structure of the fibroblast, so-called cancer-associated fibroblast. CAF cells are also constituting the big part of tumor chunk. This special structure is so-called tumor microenvironment. These cells are our immune cells. Immune cells are classified into two parts. One is the myeloid immune cells and the other is the lymphoid immune cells. And in lymphoid, there are T cells and B cells. And in myeloid, there are macrophages and various granulocytes, including basophil, neutrophil, and eosinophils. Here are two types of cells. One is a T lymphocyte and there is a B lymphocyte. They look very similar to each other because they have a similar size and shape. And also they are originated from same ancestor, so it's not surprising to see that they look very similar to each other. However, their function is quite different. B cells generate antibody, and then T cells, they can attack cancer and also the infected bacterial cells. Now, even though they have different functions, they look similar, then how can you distinguish whether they are really different cells? In this case, we utilize the specific protein expressed on T cell and on B cells. If this protein is expressed only in B cells, we may call that protein as a B cells specific biomarker. If we get this kind of a B cell specific the biomarker, then one approach is we can generate specific monoclonal antibody to this biomarker. And this antibody, if it is labeled with a fluorescence molecule, then this whole antibody is a B cell specific antibody and it can recognize the B cell biomarker. Then this antibody can be the probe which can distinguish and detect the biomarkers. So biomarker is the partner inside the body, and probe is artificially prepared. We put into the biological system to recognize the biomarker. Again, probe is the partner of a biomarker, and probe is what we enter into the biological system. Biomarker is the partner waiting inside the biological system. If that kind of a biomarker is on cell surface, we call those biomarker as a CD. It's not compact disk. It is called as a cluster of designation. So this biomarker can be specific for only one type of cell. If it is the case, then by preparing its monoclonal antibody and fluorescently label it, then this antibody can recognize this whole cell through this specific biomarker. Now, let's say if this biomarker is cancer specific, then we can call this biomarker as a cancer biomarker. Now, one cell can generate thousands of different proteins, and then if you isolate those proteins through two-dimensional chromatography, and this whole collection of proteins is what you call proteome. Out of this complex mixtures of a protein, if you can find specific biomarker for cancer, then this protein can be called as cancer biomarker. If you can get this kind of a cancer specific the biomarker, one approach to attack cancer is we generate antibodies. So this antibody is a monoclonal antibody which can specifically recognize this biomarker. Then this is a cancer-specific antibody. Now, if this biomarker is functionally important for the cancer, then the antibody itself can be a therapeutic agent. It binds to the specific protein. It can be receptor or it can be enzyme. Then it can inhibit its function of cancer. Then it can kill the cancer. Or in other case, maybe this biomarker is specific to the cancer, but it is not directly related to the function. Still, there is a possibility. Now, instead of a fluorescence molecule, we can attach this drug molecule to the antibody. Then the antibody can recognize the cancer cells. And near to the cancer cell, this drug is released and it can attack the cancer. This kind of the therapeutic agent is called as ADC, antibody drug conjugate. Most of those anti-cancer drugs, so-called the chemotherapy drugs, are cytotoxic to any kind of fast-growing other cells. So in this case, the specificity can be achieved by antibody part, and the drug can be delivered only to the cancer cells and suppressing the general toxicity, but it can kill effectively the cancers. We know that the antibody is generated by B cell. And we said B cell and T cell is part of an adaptive immune system. That means comparing to other immune cells, which does not recognize the specific structure of an antigen, and these immune cells, like macrophage or the granulocyte, they generally attack the invaders, but it does not recognize the specific antigen. But B cell does it through antibody. Then how about the T cells? T cell is also part of an adaptive immune cell. Then how the T cells can recognize those specificity of antigens? So this is the structure of antibody, and this part is antigen binding part, and we have learned that this antigen binding part is the combination of different genes, and then those combinatorial power can generate millions of the different structure of binding motif. Very interestingly, T cell also has this kind of combinatorial structure, so-called T cell receptors. T cell receptor also has this kind of a combination power for those antigen binding site, so-called T cell receptor. One difference between antibody and T cell receptor is its binding the property. And the antibody can bind to a small antigen, but T cell receptor, it needs some partners. So those partners is called as a MHC, major histocompatibility complex. And then this complex has two kinds, MHC1 and MHC2 type, and then it can present antigens. So T-cell receptor can recognize those antigen only when the antigen is bound complexed together with the MHC. So that is the difference between the T-cell receptor and antibody. If a cancer cell presents its specific antigen, then those antigens are different from normal cells antigens, so it can be detected by T cells. So one strategy the cancer can use is they try to remove this MHC. Then the cancer cell's antigen cannot be detected by T cells. In that case, our body utilizes another the immune cells, so-called natural killer cells. Even though natural killer cells does not recognize specifically the antigen, but it detects the MHCs. So MHC complex is like our identity card. And then if the cancer cell does not show this MHC, then natural killer cells they perceive that this cancer cell is not part of our cell, then it attacks. So this is kind of to avoid those strategies of cancer cells, the anti-cancer the effect against the T cells. So let's look at the T cell receptors, the recognition. So this is T cell receptor, and then here is the MHC, the complex. So this is the MHC1, and then it presents the antigen here. And this antigen, the complex with the MHC, can be recognized by T cell receptor. Now here, there are two kinds of other T cells we have. One is the CD8 cell. This is the T killer cells. And CD4 cells are T helper cells. And then they recognize different kind of MHC complex. T killer cells, they recognize the MHC1 itself. And then these two binding should happen together. Then this T cell receptor can be activated. So this is T killer cells recognition to MHC1 complex, and this is a T helper cell can recognize MHC2 class and then recognize these antigens. When T cell receptor recognized the antigen expressed by MHC complex by tumor cell, now T cell is ready to attack the tumor cells. But to avoid any kind of a misunderstanding or miscommunication, there is another checkpoint. So T cell has a PD-1 receptor, and then tumor cell can express PD, the L1, ligand 1 expression, and when they recognize to each other, then this checkpoint can suppress the function of T cells. In a sense, this is another strategy expressed by tumor cells to avoid this the attack by T cells. This is a so-called immune checkpoint. To avoid this immune checkpoint, one possibility is if there's any inhibitor of binding of this PD-1 to PD-L1, then this binding is broken, then the T cell is activated and attack the tumor cells. So this is one kind of utilizing the active immune response to attack the tumors. In this case, maybe the drug can be directly attacked to the PD-1, or it can be bind to PD-L1. Either way, it also works. Through this new immunotherapy the drug based on the antibody structure, several years ago, the previous the president, the U.S. President Jimmy Carter's melanoma was cured even at the age of 91. And this new idea of boosting the immune system through immune checkpoint got the Nobel Prize in 2018. So the starting point was PD-1 and PD-L1. Now there are many other the immune checkpoints are under study to develop the more the various and the more powerful the cancer drugs. Above antibody, even the whole cell can be used for therapeutic agent for tumor. So here is the representative example of modified T cells, so-called CAR-T. So here the CAR is chimeric antigen receptor T cells. T cells has its own the receptor, so-called T cell receptor. And then if this T cell receptor can recognize the antigen on cancer, then it should attack the cancer and then destroy the cancers. But sometimes the recognition of this T cell receptor to the antigen is not specific or strong enough. In that case, we can help these T cells. How can we help them? In T cell, this is inside the T cell and this is T cell receptor. T cell receptor is similar to antibody structure we said, but sometimes maybe that recognition is not specific enough. Also, it requires the co-complex with the MHC, so there's some limitations of this T cell receptor's function. In that case, one engineering method is possible by replacing this T cell receptor with antibody-like structure. Single chain FV was the part of the antigen binding motif of antigen and making it into a single chain. So by replacing this T cell receptor with the antibody-like structure and also activating motif inside the cell, then the modified cells has chimeric antigen receptor. Originally, this position is for T cell receptor, but now it has antibody-like structure on the cell surface. Now it can recognize the T cells much better and can attack the tumors. So this modified T cell approach is called as CAR-T.